of antibiotics and the mechanism of action of penicillin in particular. I like to start off with the discovery of antibiotics, the first of which was penicillin, discovered in part by the work of Alexander Fleming. Alexander Fleming had been working with Staphylococcus aureus and streaking that organism on plates to grow it and study its virulence. And in one particular plate, he happened to get a contaminating mold. And the mold grew and created this zone of inhibition between itself and the Staph aureus colony, and which immediately suggested to him that the mold was producing something that the bacterium couldn't handle. And so what exactly that was went largely unstudied for the next 10 or so years until another group of researchers, Chain and Flory and a few others, decided to dig into this, research topic a little bit more and literally dig into this agar and purify the compound, which they eventually named penicillin. It was produced just in time for large-scale production in the 1940s to make a big difference in World War II, which is kind of ironic. It was sort of purified just in time to save people from bacterial infections that otherwise probably would have died of bullets. So anyway, it's a dubious thing to say that World War II was probably the first major war in which microbes killed fewer people than the actual war itself. In all prior wars, like World War I, for example, and even the Civil War especially, it's believed that way more people died of microbial infections than actual bombs and bullets. So one thing we do in the lab to evaluate antimicrobial activity is a disc agar diffusion type assay, or the Kirby-Bauer method, as we call it. In this particular case, a pure culture of a particular organism, say something that you isolated from your throat or something like that, or say something that a doctor isolated from your throat and sent off to the lab. The organism would be inoculated as a heavy lawn across a plate that the microbe grows well on. We use a few different kinds of media for this, and all these media have a few things in common. One, they grow microbes fairly well, so they're nutrient rich, but also they don't interfere with the migration or chemical activity of the antibiotics that we're using. So after you've made a nice inoculum of the bacteria, usually by swabbing the microbes on the surface of a plate in a few directions, then you would lay down with some sterile tweezers antibiotic discs on the surface of the medium and then incubate the plate, and then you can measure these zones of inhibition that may or may not result. Sometimes they're very small or hard to detect or none at all, and usually they're various lengths, and so we would measure these in millimeters. And so for example, imagine if this was amoxicillin and this is a Staphylococcus aureus. The zone of inhibition is 38 millimeters, and so we would go to our table, amoxicillin for staph, and basically ask the question, where does that 38 millimeter zone fall? The organism is either resistant to the drug or some intermediate or is susceptible. And so in this particular case, because our zone of inhibition is 38 millimeters, the organism is susceptible to the drug, because a zone that's 20 millimeters or more equals susceptibility. And so again, just having a zone isn't enough. You have to actually compare it to this table in order to really understand what that zone means. So some of the desirable characteristics of antibiotics, and this list is pretty outdated, I would say, in some ways. The first one, wide spectrum of activity. Hopefully you understand why that's a little bit outdated. We wanted to kill or inhibit many different species of pathogen, and that was the original plan. If you were a pharmaceutical company, the ideal drug would have a wide spectrum of activity, could cure, gonorrhea and syphilis and chlamydia all at once. But often these wide spectrum drugs will inhibit or kill lots of microbes in your gut, and so that can be a problem. obviously these days we're very in tune to the role of probiotic microbes, especially those in the gut. We always want the drug to be non-toxic to the host and without undesirable side effects. And so that's a very important one that will never change. Obviously non-allergenic to the hosts. Penicillin allergies, for example, have limited the effectiveness of penicillin to some extent. So obviously we want them to be non-allergenic or minimally allergenic. And these two kind of go hand in hand. They should not eliminate the normal microbiota of your gut or skin or wherever. They should reach the site of infection. So obviously the drugs should not be inhibited or destroyed by some components of your body. So some drugs, for example, can't be taken orally because they're extremely pH sensitive, and those then have to be injected. And so again, it has to be able to reach the site of infection. They have to, well, not have to, but ideally should be inexpensive and easy to produce. So not requiring an extensive, a bunch of chemical purifications and so forth that would add to the cost. They need to be chemically stable, and especially if they're going to be delivered and used widely in third world countries where refrigeration isn't as common. And then, a really big whatever wish list feature would be microbial resistance is uncommon or unlikely to develop, which again, that's very wishful thinking. So some of the antibiotic targets. Now, lots of drugs interfere with replication of DNA, and these include like quinolones and rifampin type drugs that will interfere with DNA gyrase or polymerase that would facilitate replication of DNA. Rifampin actually interferes with RNA polymerase, so it interferes with transcription. So the usage of DNA as a template to make a messenger RNA copy. And so quinolones specifically interfere with DNA gyrase, for example, and certain nDNA polymerases. And rifampin specifically interferes with RNA polymerase. A lot of the drugs interfere with protein synthesis. So chloramphenicol, erythromycin, tetracycline, and streptomycin all bind to some region of ribosomes to interfere with protein synthesis or translation. And so a microbe then could modify its ribosomes in order to become resistant to one of these drugs. A lot of the drugs, like penicillins that you're familiar with, interfere with cell wall synthesis. And so they interfere with microbes being able to assemble their peptidoglycan. And if the microbe isn't growing and it's not assembling peptidoglycan, then the microbe probably won't be impacted by the presence of that drug. Some drugs work on enzymatic activity, such as the sulfonamides and trimethoprim, which interfere with very specific enzymes in certain biochemical pathways, like, for example, folic acid synthesis. Microbes absolutely need folic acid to produce DNA nucleotides, for example. And so without that folic acid synthesis pathway, the microbes can be inhibited. And so trimethoprim and sulfanilamide type drugs would interfere with that, as an example. Polymyxin B, which is an over-the-counter, often added to those topical creams, interferes with plasma membrane functioning, but more specifically with a lipid that carries peptidoglycan across the cell envelope, and so as a result, the microbe can't properly assemble its cell wall. So here's an image of penicillin in action. You see these microbes growing, but their cell wall is weakened by the action of penicillin. They become osmotically sensitive, and then they burst. And so I say here, see animation. So in this particular case, here's the peptidoglycan. Here's these repeating NAM and NAG subunits, as I've talked about, and then these amino acid side chains. And these side chains have to be linked together in order to solidify the cell wall. And the enzyme that does it is called transpeptidase, as you see here. So transpeptidase will come in and remove the terminal alanine. You see it going away there, and then and then adds a bond between the two side chains. So there's the bond that it added. I don't know if you noticed that. Go back and you can take a look. So this terminal alanine goes away, and then a bond is made between this alanine and this amino acid, this unusual amino acid called DAP, or diaminopamelic acid. So transpeptidase comes in, forms the bond, plucks off the terminal alanine, and then you have a strong cell wall. But penicillin actually will come in and bind to transpeptidase and prevent that from happening. So some of the mechanisms of resistance then, whoops. So the mechanisms of resistance then, as we can see here, summary of resistance mechanisms, include a few different things. So number one here is a altered antibiotic target. So that's something that I had alluded to before, where imagine you have a protein synthesis inhibitor that would bind to ribosomes, and that protein synthesis inhibitor will bind to the ribosome and prevent translation of messenger RNA into protein. But the microbe can actually alter the ribosome so that the antibiotic no longer binds to it, and then will allow translation to proceed even in the presence of the antibiotic. Another type of approach that microbes use are antibiotic degrading enzymes. And so they actually pump out an enzyme into the world that chops up the antibiotic into little bits, and so that it doesn't work. And then the third type over here is a subtle variation of that previous theme. This is an antibiotic altering enzyme. So the antibiotic is largely unchanged except for a few functional groups that are added. And so a methylation, for example, or a phosphorylation or an acetylation. So a minor functional group is added so that the antibiotic no longer works, which is a bit different than actually just chopping it to pieces. And then another version of this is the antibiotic actually comes in, but the microbe has an efflux pump that the microbe will use energy or proton motive force to pump the antibiotics out. And then this last slide just shows origin and spread of resistance genes. How do microbes acquire these, plasmids, for example, that encode all these different activities? And so one way they can do that is through conjugation. And so here's a donor bacterium that has this multi-drug resistant plasmid, and the microbes can actually mate through a mating pilus, where this plasmid is transferred through a pilus into the recipient. Now this recipient becomes resistant to the drug. Another way is transformation. So imagine this neighboring cell in this complex consortium breaks open, lyses somehow, and releases its DNA into the medium. And the microbe, this neighboring microbe, can then acquire pieces of that DNA and potentially recombine them into its genome to make the microbe resistant to certain antibiotics. And so that's called transformation. This is conjugation. And then lastly, another version called transduction, in which this bacterium is infected by a virus, and the virus packages bits of the host DNA into its capsid, and then the virus goes off into the world and injects that previous host's DNA into the recipient. And so one version of things that can happen here is that this virus can transfer this guy's antibiotic resistance genes to this recipient. This process is called transduction. And so once that transduced piece of DNA enters, it can integrate into the genome and allow the microbe to be resistant to that particular drug.